All right, it's 11:01. We're going to wait just a minute or so and allow everybody to log in and get properly situated, and then we will get started. Okay, a few more people logging in here. Looks like it's leveled off, so we'll go ahead and get started. Good afternoon, good evening, and good morning, everybody, depending on where you may be logging in from around the world. I am Rob Burgess. I head up business development for Sinobiological, and it's my pleasure to welcome each of you to the next installment in our Lock and Key series of live and, of course, free webinars, which focus on cutting edge technologies as they apply to therapeutics development, diagnostic kits development, and even basic research. We have a fantastic speaker today going to talk to us about some bioluminescent technologies. I will introduce the speaker here in just a second. I did want to mention one housekeeping issue, and that is, if you don't mind, please log into the chat box, say hello, tell us where you're from. We're always interested to see where people are calling in from around the world. And if you have a question for the speaker, please ask that question in the chat box. And at the close of the webinar today, I will walk through all of those questions in the chat box and get to as many as possible by verbally asking those of our speaker. So thank you for doing that. Do please tell us where you're from. And with that, it is my pleasure and honor to introduce everybody to our speaker for today. That's Dr. Hicham Zegzouti. Hicham is a long-term friend of mine and a great colleague of mine. We've known each other for many years. And I'm just going to read out his profile because it's so extensive and I want to make sure I get it right here. Dr. Zegzouti is a senior research scientist and a group leader at Promega R&D, where he leads the development of innovative biochemical and cell-based assay technologies which target kinase signaling, targeted protein degradation, and epigenetics to support small and large-scale drug discovery. Hicham got his doctorate in molecular and cell biology in 1997 from the National Polytechnic Institute of Toulouse, and this was followed by a postdoctoral research program at UC Berkeley as well as UCLA, where he explored AGC kinases and hormone signaling in Arabidopsis. He's been with Promega since 2007, and there he's played a pivotal role in advancing and commercializing assay platforms that enable cancer research and therapeutic screening. And this particularly applies to that for kinase biology. Hicham has authored over 30 peer-reviewed publications and patents, and he actively promotes cross-functional collaborations and is a speaker at many different scientific conferences and contributes quite significantly to Promega's assay innovation strategy. And the title of Hicham's talk today is Bioluminescent Assays to Analyze Signaling and Degradation Dynamics. And with that, Hicham, it's a pleasure to have you today, and I will turn the platform and virtual podium over to you.
All right. Thank you, Rob. Thank you for the introduction and the kind invitation to give this talk. Yes, as you said, we know each other for quite some time now, and hopefully, our friendship and partnership will continue for many, many years.
Indeed, it will.
All right. So today, my talk will be like centered around exactly what the title is saying. I'm going to be talking about some bioluminescent assays that we developed over the years. And to start with that, so I'm going to just set the stage of why we are doing all that. So as all you know that many mutations of the proteome actually rewire signaling inside the cell, and that drives disease phenotype. So everything we talk about today is basically disease, targets, drugs, and assays. Of course, that is the main a subject how to interrogate the activity of drug target or level of drug target in cells or biochemical in a biochemical way. So I put an example of families of drug targets that we call PTM enzymes. It's a name that we invented. I don't know if really we call them PTM enzymes, but as the name says, it's enzymes that actually their role is to do post-translational modifications. And usually these are large families. You have kinases, you have ubiquitin ligases, methyltransferases, etc. And those are large and they drive a lot of biological events in the cell. And of course, if there are mutations in those proteins, that actually drive the disease state of the cell or of the tissue. So this is actually something that we start with, or pharma companies start with, or anybody who is studying targets or proteins that give disease. Usually, it goes without saying that it has to be after identification of the target, you have to validate, you have to link the genotype or the biology to some tractable intervention using different assays or multiomics or perturbation studies to validate that target is the one driving the disease phenotype. And of course, when you do that, you are looking for, inhibiting or degrading or preventing that protein or target from misbehaving. And to do that, you have many therapeutic modalities that can act on those targets. And here I'm just showing like the three major ones that I'm going to be describing assays for, and also some case studies that we did here at Promega. So, of course, small molecule inhibitors are the ones that a lot of programs are based on. So you have small molecules that bind active targets and preventing them from, doing their work. And this, of course, because you have overexpression or you have higher activity of the protein. Here it's a kinase, and the drug will prevent it from phosphorylating its substrate. Now, 10 years ago or more, there is a new modality called target protein degradation using PROTACs or molecular glues. This is mainly, or we can say it's been used really successfully with undruggables, like proteins that don't have pockets where you can fit those small molecules. And this is, you have two types for now, PROTACs and molecular glues. This is to drive the degradation through hijacking the ubiquitin proteasome system. And that actually is something that is a lot of people are working on. And I will show you some example later on. And of course, you have also large molecule drugs, and those are mainly antibodies that bind either ligands or receptors outside the cell to do similar thing, which is to inhibit pathways or to drive the degradation through, the biological degradation of those receptors, for example. So those are three modalities that I will show some assays for today. So to discover and optimize these modalities, of course, you need sensitive and robust assays that can be applicable to high throughput screening or high content screening and MOA studies. And at Promega, we built like a large array of biochemical and cell-based assays to either quantify the target activity, protein level, including degradation, and pathway inhibition and activation with some HTS grade performance. So in my talk today, I'm going to describe two sets of assays, but I will mainly focus on the new generation of cell-based assays called Lumit. But just I will introduce or remind people of the biochemical platform that we have. and everything here is bioluminescent. So early on during my tenure at Promega here, we've been interested in looking at drug targets, and we started with the kinases. For example, as you can see, the kinases use ATP and generate ADP. Many of the drug target families do similar things, but they generate either a nucleotide that can be ADP or can be actually converted to ATP that we can use bioluminescence. So we couple every nucleotide conversion with an enzyme that will convert it to ATP. And in the presence of firefly luciferase that uses ATP, we will have exactly the level of production of that nucleotide. So, for example, ATPases or kinases will use ATP, will generate ADP. We'll just convert ADP to ATP, and then luciferase take care of luminescence. Other drug targets, we found a way to do it also through coupled enzyme reaction, and we have built a successful toolbox for various enzyme characterizations or high throughput screening. So I'm not going to go through these because there are a lot of publications around them. I will just give you the example, the major example, which is ADP Glow. We all call them Glow. Which is being used for kinase activity detection, ATPase activity detection. We published a lot of papers on that, and many others also did. So the way this assay works, and that's the example I'm giving here, you can see that after the kinase reaction or the ATPase reaction, you have production of ADP after a substrate phosphorylation, and you have ATP that is left in the reaction solution. So we have a one step. The first step is called ATP depletion. We add a reagent that gets rid of that ATP, and then we add the second reagent that converts ADP to ATP, and then ATP will be used by luciferase to emit light. And this assay is very powerful, very sensitive. You can use very small volume for HTS, so it can be used in high throughput screening. A lot of mode of action studies have been done with this assay, and we have actually a partnership with Signal Chem, part of Sinobiological, where we actually provide over 370 kinase enzyme systems, which are kits of kinases or kinase mutants that have everything you need: kinase, substrate, buffer, cofactors, etc., that is ready and validated with ADP Glow. And this partnership is going for a long time now, which is great, and we're trying to continue that for the next future. So I'm not going to stay on the biochemical assays. There are a lot of publications. Just here, it's to show you that we are covering basically many cell signaling pathways. As you can see, we can detect, cyclic GMP, cyclic AMP. We can detect kinases, demethylases, epigenetic enzymes, etc. And many publications are there. You can access them. You can actually read about these technologies over the last 15 years that we have. So I'm going to switch gear, and now I will talk about the new assay called Lumit, which is a new bioluminescent immunoassay. And before I do that, I want to actually just remind you, if you don't know about our nanoluck technology. Nanoluck is the new generation of small luciferases that are bright, and it was derived from deep sea shrimp. And through molecular evolution, we made it very bright. And as you can see here, this image is taken actually with a handheld iPhone on the plate to show you that it's a very bright enzyme. So this enzyme is being used as a reporter for many biological assays. But what we did, we took also nanoluck and we cut it into two pieces. As you can see here, you have a large fragment called large bit, which has 17 kilodalton. and then the C-terminal subunit, we call it subunit, it's just the last 13 amino acid of the enzyme. If you separate them, what happens is you have very, very low affinity between the small fragment and the large fragment, or we're going to be calling them small bit and large bit. They have very, very low affinity, 190 micromolar Kd. So in solution, these two pieces don't interact. However, when you actually tag or recombinantly create two proteins, one with the small bit and one with large bit, and if those two proteins actually interact, that brings into proximity the large bit and small bit, and that reconstitute again the nanologue and creates light in presence of the substrate. And that's where Lumit comes in. We took advantage of this nanobit technology that has been used for PPI, for many studies, and we label antibodies with the small fragment and the large fragment, small bit and large bit. And in the presence of an analyte where those two antibodies recognize two different epitopes, the antibodies actually will bring into proximity the two small bit and large bits, and that generate light. So it's a homogeneous assay. We took actually a traditional ELISA workflow, which is, as you can know, complex and slow with a lot of washes and steps and additions, to a very simple assay where you have the analyte addition, you have the addition of either the primary labeled with the small bit large bit, or primary that are not labeled but secondaries that are labeled. You incubate and then you add substrate, and you go to a luminometer and you detect the light. It's very easy assay, fast. It takes some assays takes 30 minutes, some assays take 2 hours, but it's very fast. It has high sensitivity, and it has a broad dynamic range, and it's flexible because it's homogeneous. So you need to just add, and read. So it's very applicable to HTS platforms. So there are many concepts of Lumit. As you can see here, I put many of them. You have the direct, which, as I said, you can label primary antibodies. We have kits that can help you do that. You have the indirect, and that's the one I'm going to describe because my team was in the development of the indirect assay because we wanted to develop kinase assays, of course, and we couldn't create assays with the direct because you have so many. So we created a platform where you have secondary antibodies that are labeled, but it's a plug and play. We just, for every target, we just switch the primary antibodies. You can use anti-tags for protein interaction in a biochemical way. So you can have two proteins, one with the GST and one with His, and you can use an anti-His and an anti-GST, and then you emit light, of course, like Lumit does. So, like I said, the direct, we have an assay or a kit to label. We developed a lot of cytokine assays that are available. The indirect, that will be our talk today about phosphorylation and degradation. So we'll go through it. The anti-tags, as I said, we have like 6 or 7 anti-tag antibodies that can be used in a biochemical detection of PPI. And then, of course, you have competitive binding, where you don't label actually two antibodies. You have one antibody that is labeled with large fragments. And then you have the analyte that could be a toxin, could be a second messenger. So where you cannot have two antibodies. So you label that, and it's a competitive assay. And then during COVID, we created serology assays and RBDase2 PPI assays. So those are like published also that you can look at them. So today's talk, we're going to be talking about Lumit immunoassay cellular systems, and that's the one that uses secondary antibodies that are labeled. And for just saying it quickly instead of a long sentence, we're going to call it Lumit ICS. So I'm going to describe some Lumit ICS for phosphoprotein detection, for target protein degradation, and also for HTS. Okay, so the first thing is, how does this assay work? Imagine you have a cell, you have a pathway that is activated, you want to inhibit it, or you just want to activate it. After treatment of the cells, you just add a lysis buffer and incubate for 40 minutes. And then what we do is we add actually 4 antibodies at the same time. 2 primary antibodies against the target. So here, as you know, we need to have 2 different species, rabbit and mouse, or mouse and rabbit and rat, etc. We have many variants. One that recognizes the phosphorylation, and the other one that recognizes another epitope. And of course, you're going to have an anti-rabbit that is labeled with small or large bit, and the anti-mouse again with large or small bit. And what you do, you just incubate this for 60 to 90 minutes, and you add the reagents and you detect luminescence. So here for the phosphoprotein detection, I'm going to take it as an example to show you how this assay is specific. We call it a two-factor authentication because if you have different like antibodies that have some non-specific binders, so they bind other things, for example, you will not generate any light. You generate light only when all the complex is formed with the 4 antibodies. It means you are detecting specifically the phosphorylation on the target of interest because you have the other antibody creating the specificity. In a Western, for example, where you have non-specific antibody binding to different proteins, you will see those proteins on the gel. Same thing for total protein. If you are studying total protein level, either in a drug, discovery, you want to degrade the protein, you're going to see its degradation. It's the same way. You just now have the 2 antibodies are recognizing 2 different epitopes outside any modification like phosphorylation, and now you are looking at the total level of protein. So very rapid add and read formats to interrogate any cell signaling pathways or looking at the level of proteins. One particular thing that we added into this assay is adding a multiplexing or duplexing with a fluorogenic compound. So this is our cell titer fluor, which is a compound, glycine phenylalanine AFC. This is not fluorescent. It enters the cell. So if you add it 30 minutes before lysis, it enters the cell, and you have an active protease in the cell that start showing the GF and liberating the AFC, which is starting now to show fluorescence. So the fluorescence will accumulate for the 30 minutes before lysis. And once you lyse, now the fluorescence accumulation starts, and you can read the fluorescence and luminescence at the end and do normalization to the cell level, to look at the cell viability if your compound is killing the cell, etc. And I will show you some examples also. So again, this is a homogeneous assay, add, and read. And like I said, we have antibodies, anti-mouse, rabbit, and rat that you can combine with different primary antibodies to create an assay. And we have also assays that we created. So these are like over 50 phospho and total target that we looked at, and we created app notes that help actually whoever wants to develop the assay to locate what primary antibodies to get, etc. So as you can see, many of them are phospho targets. You have phosphotyrosine kinases. You have some total level or total protein detection, some drug targets for PROTACs, etc. And now, because we are trying to move to the next level where we have complete kits that contain also everything. So we are creating some assays that are relevant to cell signaling or TPD, and those are available also for people to use without any optimization. Okay, so my first case study is the work that we've done with the phospho-ERK. So what we decided is to look at node signaling. So the node signaling is one node like ERK or phospho-ERK can be a reporter for many events that happen upstream. As you can see, we can look at different RAS MAPK pathway activation or inhibition. So we can use it to look at EGFR inhibitors. We can look at SOS RAS inhibitors, RAS inhibitors, RAF, and MEK. So one assay can look at every aspect of the RAS MAPK pathway. And that's what we did. We actually validate that by activating the pathway using AGF or PMA to look at the level of activation. And that we look only at phospho-ERK at the end. And we also did inhibitors of the different targets that I just described to show really IC50 for each of those kinases or RAS inhibitors. But the main work was to look at ranking or rank order the drugs. At that time, it was the beginning of the RAS inhibitor saga, and we took those inhibitors and we looked at them. But before we did that, as you understand, every time we publish some study using our Lumit, the reviewers ask us about Western. How does it compare to Western? As you can see here, we did this experiment where we looked at PMA activation of the pathway. We look at phospho-ERK in different number of cells, and we inhibit it also with trametinib, which is a MEK inhibitor. And as you can see, of course, you see it with the Western, but however, with Lumit, we have a very sensitive and also a very linear relationship in terms of signal. So, of course, Western is very cumbersome. If you use Lumit, you have just add, and read. But it validates our assays this way. So what we did, we took actually 4 cell lines, and these cell lines have different RAS mutations. As you can see, this MCF7 is the wild type. This one has the KRAS G12V, G12C, and G12D. And what we did at that time, we had only this inhibitors of RAS. None of them actually inhibit the wild type, of course, but we had many of them that inhibit the KRAS G12C, which is in MIA-PACA. And as you can see, AMG 510, which is the first approved drug against RAS mutation, it's inhibiting the pathway. And of course, phospho-ERK is inhibited. And we looked at the KRAS G12D, and we had the only MRTX 1133, which is G12D mutation. So we did a couple of work here to show different rank orders. And as you can see, we have also this MRTX 1133 inhibiting a G12C mutation with a higher concentration, of course. So we did a titration of those compounds in those different cells, and we can see that all the G12C are inhibiting with low IC50 the phospho-ERK. However, when we look at the G12D mutation with MRTX 1133, we can see that we start inhibiting also the other mutant in the other mutant cells. Of course, nobody will use that as a drug in terms of inhibiting because you have only one mutation when you have the disease. But at that time, it was just for us to show that MRTX 1133 can be a starting point to look at G12D and G12V mutations as a drug target. So in that paper also, because we published this work, we looked at an available PROTAC at the time, LC2, which is based on MRTX 849 as the warhead. It was published. It's a degrader of KRAS. So we did degradation of KRAS, but we didn't look at KRAS. We looked actually at phospho-ERK. And you can see that the degrader actually inhibited, of course, the pathway, and we can see through phospho-ERK. And this is the IC50 for that LC2. Like I said, this is something that we are looking at as a homogeneous assay, so we want to have it as HTS compatible. So we looked at miniaturization in 384, and of course we do a lot of optimization to make it compatible with 384. And we have also a collaboration. So this is just a Z factor that we did for a small screen in 384 using phosphor-ERK again. And we identified all the compounds from this library that have an effect on the pathway that leads to phosphor-ERK. We also have a collaboration with Novartis in Cambridge where they did 1536 optimization. We looked at the MOA box library that they have, and we found the different activators and inhibitors of the pathways. And this work actually has been validated, and we are right now writing a paper. It will hopefully be submitted before the end of the year, and it will be available hopefully next year. So I talked about small molecule, HTS. Because this is a cell-based assay, you can also look at the effect of large molecules on signaling by looking at different nodes or looking even at the phosphorylation of the receptor. So this is the example of phospho-EGFR immunoassay using Lumit technology. Just a quick description here. We use an anti-EGFR antibody that recognizes extracellular domain of EGFR and one that recognizes the phosphorylation. After EGF stimulation, of course, you have a phosphorylation that brings together all the complex, and that forms light. And here is the validation. As you can see, we can look at different phosphorylations here. And we even looked at a pan-phosphotyrosine phosphorylation or antibody. And as you can see, it gives also similar results or IC50s. Now that we have a receptor tyrosine kinase assay, we wanted to look at the effect of monoclonal antibodies or biologics. Here is cetuximab against EGFR. It's a titration. We look actually at the IC50 looking at phospho-EGFR inhibition. And of course, we did a lot of validation to look at the antibodies that we add do not interfere with the assay. So we did those tests, it's just I'm not showing them here. We looked at different biologics for that. But what I want to show you today is how to study actually large and small molecules using similar assay. So this is just a quick study that we did. We took HCC 827 cells that have an EGFR activation deletion. So we are starting already with a phosphorylation or activity of EGFR. It means it's highly active. So if we add a small molecule like erlotinib, which inhibit the kinase, you get like a result like this. You start from, let's say, the basal level, which is high already in these cells. You have 1 million RNUs, and that, of course, it's inhibited, as you can see from this picture. Now, this we call it the basal phosphorylation level. Now, if you add a biologic to block EGFR, nothing happens. So cetuximab actually doesn't do anything because it blocks only the receptor ligand interaction. And then to see actually that effect, we added EGF. And when you add EGF, you have an increase in phosphorylation. And it's shown here by the increase in basal level. But of course, if you add erlotinib, you're going to inhibit both the activated and the basal level. So you go down to zero. And this is now where you can see the effect of the biologics. So the biologics prevent the ligand from binding, and that create an inhibition of the ligand-stimulated EGFR, but not the basal level. So it goes back just to the basal level. So this is a kind of showing what goes through, the basal activity versus the activated activity of the receptor. We looked also at HER2, which is one of the other receptors or the EGFR family. This one doesn't have a ligand, of course, that is known. So we could have a cell line that has a high activity that we can inhibit with a compound that inhibits the HER2 kinase activity, that CP compound. So that inhibits. But then when we add a biologic, trastuzumab or pertuzumab, that bind the receptor, actually we see an increase. And this increase actually is known because at low concentration of the biologic, it works like a ligand for this. You have to have a higher concentration to prevent actually the dimerization of HER2 with other receptors to see the inhibition. So we looked at this early stage activation with the biologic, and then we want to compare that to an ADC based on the same trastuzumab. So as you can see, the compound again is inhibiting the HER2 phosphorylation, but the ADC is activating also the same way. So now we are staying just at the receptor level. But if we go down and look at some nodes of signaling, we looked at AKT phosphorylation by treating with the biologic or the ADC, the antibody drug conjugate. As you can see, AKT phosphorylation is inhibited downstream with the biologic, but the ADC doesn't have much of an effect. So we are thinking that the anti-HER2 antibody reduces AKT signaling because that actually drives uh, the proliferation. So we're inhibiting actually the proliferation through AKT. But the ADC doesn't, because ADC is actually goes through toxicity through the DNA damage. And we wanted to verify that. And this is another assay that we have developed for gamma H2AX. This is a marker, a phosphorylation marker of a histone 2AX that is increasing during DNA damage response. And we can see actually that trastuzumab doesn't do that, but the ADC does. So these are the kinds of studies that we look at just to validate our assays and to show some biologically relevant events. Okay, so this is the last part of my talk. I'm going to switch now to degradation, looking at, of course, small molecules, but now it's small molecule degraders. Promega has invested for a long time now to creating technologies that interrogate the TPD field. So here, like you can see, PROTACs, they form binary and ternary complex with the E3 ligase and the target. Then you have ubiquitination of the target. Then you have proteasome degradation. So Promega has a lot of technologies using our Halo tag, nanobreads. Probably you heard about them. So we look at if the permeability of the cell allows the PROTAC to go in by looking at the nanobreads, target engagement of the PROTAC with the E3 ligase. We look at ubiquitination, looking at nanobreads using a Halo tag, ubiquitin, and then the target labeled with nanolock. We look also at if the PROTAC actually forms the ternary complex that is the start of forming that tight complex that can be used to recruit the ubiquitination, of course, through the A3 ligase. So all of these parts we have assays for. And then the last one, we have the high-bit technology. I don't have time to go through it, but quickly, high-bit is like small bit, but the small bit is, as I said, is very low affinity. It means you need to have some close proximity to form the nanobits and light. High bit, we call it high affinity. It means from the get-go, if you have a cell line that have a target with the high bit and you add large bit, quickly they find each other and they show light. It means it's a detection module where you can actually CRISPR the small 11 amino acid tag onto any target you want, and you can follow its level or degradation through, PROTAC or molecular glue. So this technology is becoming really the go-to technology for looking at uh, degradation of the target. And as you can see, you can create really some kinetic studies to look at different time course for degradation of your target. Here, for example, BRD3. So there are also some papers describing this technology. Of course, this needs engineering cells with a high bit and looking at the degradation of the target. However, if you want to look at non-engineered cell models, you know, Western has been used extensively for that to look at degradation. Here is a BTK degradation. But that's where Lumit comes. And I'm going to show you how Lumit can replace these cumbersome technologies because it's quick and easy. And also it has some advantages where you can do quickly profiling different cell lines because there is no requirement of engineering. So here is the assay. It looks like a relevant cell. You want to degrade this protein target. When you have no treatment, you have the complex form that forms luminescence, and then you add your degrader, you don't have any luminescence. And I'm going to show you how we use that for some targets. So we developed a lot of assays for different drug targets. As you can see, the green ones here are the ones that I'm going to show today. We have some neo substrates and some drug targets like SMARCA2, STAT3, BRD4, BTK. We validate always our assays by looking at proteasome inhibition to show that that this degrader goes through the proteasome. And also sometimes we add an analog of the compound or the warhead to show that it doesn't have any effect, only the PROTAC has an effect. We developed also different neo-substrates. The neo-substrates are very molecular glue specific because they get degraded with the cereblon E3 ligase. So we have assays also for those, and I will show you how we use them. Quickly, we compared Lumit with high-bit technology, and as you can see here, we get the same DC50 for here SMARCA2 or SMARCA4 using their corresponding degraders. You can see that the DC50s are exactly the same. We use this technology also to look at time course. Even if we cannot do kinetics in the same well, we can do them in different wells. So we looked at 6 pairs like a target and degrader, and we looked at their degradation. So you have some that are really fast, like here ARV771 with BRD4, and you have some that are slow. You have to wait until 24 hours to see a decent degradation. By the way, here I'm showing you exactly where we use that GFAFC. So you can see that over time we look at the cell viability of the target, cell viability of the cells that were treated, and you can see it gives you an idea that your cells were still healthy. Only the target is degraded, and that's why luminescence is low. We look also at DC50 of those degradations, and here again we use the GFAFC compound to look at the cell viability and to normalize also. So one little study that we did for on and off target degradation, of course, we looked at one which is STAT3 with SD36. Here we compared the degradation of STAT3 versus the degradation of 5 neo substrates that are prone to be degraded by cereblon. And you can see that this degrader, even if it goes through cereblon, has no effect on the neosubstrates. However, when we look at a different target with two different compounds, both of them go through cereblon. As you can see, this one, at 3 hours, we don't see much degradation of BTK. So BTK here is degraded with the DD compound, and the other targets are not. However, at 24 hours, you start seeing really degradation. So there is some kind of off-target degradation of these. And now, if you look at the MTA22, which is really nice and selective, you have to have really high concentration before you can see any activity. So this has taken now allow you to do quickly multiple cell lines, multiple targets at once to look at on and off target of your degrader. The last part of this degradation study is, like I told you, the degrader potency profiling can be done across diverse cellular contexts without any cell engineering. And here we just wanted to show an example for STAT3 degradation in four cell lines. And we were surprised to see that for A431, you have actually a very high DC50, and that let us think what is happening there. So we went actually and looked at 12 other cell lines, and still A431, that's the only one that has the highest DC50. So we wanted to investigate why this cell line has that barrier actually to SD36 degradation. So we did a bunch of work that was negative because we didn't find that there is any mutation on STAT3 that shows that can explain that. Cereblon or STAT3 levels where there was no correlation. We looked at Western and mass spec of those mass spec data. We looked at cell permeability. We didn't see any difference. Both actually cell lines, like if we compare A431 or K562, this compound has low permeability in both. So we did a bunch of things to find no indication of what is happening in A431 until we start looking at different things. We found a paper that came in 2024 that described that there are some pathways like PERK pathway and the PARylation pathway that could be involved in creating some kind of resistance to different degraders. And they showed that for BRD4 for different compounds. So we said, probably there are some signaling pathways that could be inhibited or activated in those cell lines, allowing resistance or potency of the compound. So we went and did the treatment for that PARylation or deparylation inhibitor. And we found, yes, there is actually similar to what they showed in the paper. When you treat with this compound, that's A431, which has the highest DC50, as you can see, the micromolar level, we can actually shift the DC50 back to low nanomolar. The PERK pathway has a little effect, like they showed, but still for us, we found that there is probably something more in A431 that is having an effect. We did actually some mass spec data comparing 26 cell lines in terms of expression of different proteins. Here is the example of EGFR. A431 is known to have high expression of EGFR, and it shows here. So we said, is there a correlation between high expression of EGFR and low DC50 for this compound? And we found that there is some correlation between the highest EGFR expression and the highest resistance. So now we are investigating the EGFR story in terms of third pathway that could prevent some PROTAC degradation. So just to show you this, now we have three pathways: the PARG, the PARylation pathway, the PERK pathway, and EGFR. Why? Because we treated actually uh, the A431 with gefitinib, which is an inhibitor of EGFR phosphorylation. And you can see we can shift back to a certain extent the DC50. So it means there is an effect of activity of EGFR on the PROTAC degradation of STAT3. So this is very new since this year we are still continuing the investigation to confirm that EGFR inhibition or degradation can allow SD36 or the cell to be more that can be that we can allow SD36 to degrade STAT3. So I showed you that after having multiple technologies for TPD research, now Lumit, ICS, and other Lumit technologies are part of the workflow. And after showing all these different biochemical assays, now we have a new assay that can show different cell signaling pathways activity that you can study with these bioluminescent technologies. So to summarize, we created a comprehensive drug target assays from biochemical to binding, like target engagement, to cellular assays using bioluminescence. Lumit is a versatile immunoassay platform for any analyte detection. Lumit Cellular System performance detecting total and phosphotargets, including phosphoreceptor tyrosine kinases, that you can interrogate biologics biology. We can actually use Lumit to look at target degraders in non-engineered cell lines, so you can do profiling of different cell lines. And also, you can adapt this assay for HTS by miniaturizing it. I would like to thank my Promega colleague. This is the team that I belong to, our director, Jim Calley. Our team is called Assay Design and Sample Analysis. These three scientists are great. They are the one who do all the work. I just have the pleasure to talk about it. They are in my team. And we collaborate with the protein and nucleic acid analysis group, with the protein degradation team, mass spec team. And also, I showed you some HTS that we did with He and Doug from Novartis. And I would like to thank you for for listening to me today. And any questions I can answer will be great.
Thank you, Hicham. That was a fantastic webinar. Appreciate it. Very, very thorough. Congrats on the development of both the ADP Glow technology, certainly the Lumit technology. It's just wonderful what y'all have done for the research community. I would like to ask our attendees one last time to please, if you have any questions, type them into the chat box. We have just a few minutes left before the top of the hour. And I will go ahead and start. Hicham, let me just ask you, and I'm sure you said this pretty clearly. I may have missed it, but you mentioned the signal chem kinases you've been working with. Can you tell us again how you've been working with those, and if the quality is good and the performance of the kinases are good? Just give us an overview of that.
Yeah, so we started around 2009 after the ADP Glow was launched. Because we didn't have kinases, the Promega, we had few. So there was like a personal relationship between Promega and Signal Chem at the time, and we talked about how about creating kinase assays that can work with ADP Glow, and of course Signal Chem has up to now really is the largest collection of kinases and kinase mutants, and it's known for the quality of the kinases. So that's what led us to talk about how about we collaborate or partner to create kinase assays that will work with ADP Glow. And of course, when we started working with them, we found about the quality, very highly active enzymes, consistent QC is being done, and we just worked a partnership where Signal Chem put everything in a box or in a bag, and they ship it to Promega, and then we ship it to our customers and single came customers too. So that's how it goes.
Sounds great, Hicham. Glad you like the kinases and the selection. Certainly, you mentioned the quality. We appreciate that partnership, and certainly
Yeah,
Promega is one of our VIP partners worldwide. So,
thank you.
um, I'm sure you probably mentioned this too, but does Promega also provide services for like the implementation of all the assays you've discussed and like readouts for customers as well?
So we have a small group called Tailored Research R&D Solutions,
Mhm.
where we do custom build-up of assays for different customers. The only service we do in terms of assays, it's mostly target engagement. We have a profiling system or service for profiling different compounds in the cell-based target engagement.
Mhm.
We don't have it for ADP glow because I think, sometimes you need to know where you are, you can bring value. And I know like Signal Chem does already profiling using
Mhm.
ADP glow and different techniques. So
Mhm.
I think we don't do that. We do mostly target engagement.
Okay, sounds great. Appreciate that. Well, I think we're going to wrap it up then. I let me just go ahead and thank the attendees. We had attendees from all over the United States as well as an attendee from Vancouver. So thank you very much to the attendees for joining us today. Hicham, that was a fantastic talk. You gave tons of data there, and I know you spent many years optimizing those assays and those technologies. So congrats once again to you and your team, and we sure appreciate you presenting today.
Thank you very much for the invitation, Rob. So we'll see you next time, huh?
Yeah, you got it. And one more person to thank, Ms. Kayla Sarmiento, who also has successfully executed yet another Lock and Key webinar series. So Kayla, thank you as well for all of your hard work. And on that note, I think we'll sign off, and I wish everybody a good afternoon or a good evening.
All right. Thank you. Bye bye.